Welcome to the Endpoints podcast, presented by the ALS Therapy Development Institute. I'm Jonathan Gang. Antibodies are an essential part of our body's defenses against disease. They are specialized proteins that attack and kill foreign invaders, like infectious bacteria and viruses. Antibodies are normally naturally produced by our immune systems in response to an infection. However, in recent years, scientists have learned to produce antibodies in a lab to treat a variety of diseases. Several antibody treatments have been investigated in ALS, including tegoprubart, formerly known as AT-1501, which was invented by ALS TDI and recently successfully completed a Phase 2A clinical trial. Dr. Silvia Pozzi is an assistant professor at the Université Laval's Brain Research Center in Quebec City, specializing in ALS research. She recently was an author on a paper reviewing the current state of antibody therapies for ALS. She joined us on Endpoints to discuss the current outlook for these therapies in the ALS space and what the future might hold for these treatments.
Let's talk about the literature review about antibody-based therapeutics that you recently published. Just to start, can you explain sort of in simple terms what an antibody-based therapeutic is?
Yeah, so basically, what is an antibody-based therapy? Well, we just take advantage of what our body generally do. So every time that we have a pathogen, we have an infection, we have something bad ongoing, the body, the immune system produce antibodies, and these antibodies have the aim, their goal is to get rid, to degrade what is produced. So an antibodies-based therapy is just to use this tool, this antibody, to target specifically a protein and then to block the effect, the pathogenic effect that this protein, this molecule is doing. And the antibodies are amazing because they can be, they are really useful. They, as I said, their nature is to get rid of the pathogen of their target, but they can also be modified. So we can use a lot of. We can modify their chemistry. We can cut them in small pieces and make them what they are called nanobodies, that they are smaller so they can penetrate more in the cells, so they can reach easily their target. They can be, there are amazing studies, and we have to learn a lot from the cancer therapy. There are antibodies that are targeting multiple cells and putting together cells, like making them closer, so the immune cells can act on the cancer cells easily. We can couple them with conjugate them with the effector molecules, and let's see. So we can really play a lot with antibodies, and but the main thing of the huge advantage of antibodies is that they are definitely extremely specific for their target. We use it normally in our experiments when we want to do an experiment and we want to search to have a look of our specific protein. We use antibodies, so we can even use antibodies for a therapy that can target a physiological protein, that can target a protein that is modified by a pathological event, like I don't know, I can say just the phosphorylation of the protein. They can block the interaction between the protein and another, let's say, a receptor or another protein, and so block the detrimental event that is induced by this interaction. So definitely a lot of advantages with these antibodies and with antibodies in general. And yes, it's worth to use them in a therapeutic approach.
And can you provide any examples of antibody treatments that are in clinical trials right now for ALS?
Yes, there are different, at least I know a couple of antibody-based therapies that treatment interventions that are in clinical trials. One of them actually comes from the ALS TDI Institute. So, it's an antibody against CD40 ligand, which is a molecule expressed by involved in inflammation. So the aim of these antibodies to block all the detrimental effect linked to inflammation that we know in ALS. And the other one is an antibody against SOD1, which is one of the most studied protein in ALS. And the idea is to get rid of the protein that has a pathological conformation of in ALS patients. So yes, definitely two good examples of antibody-based therapy that are now in clinical trials.
Good. And just tell me a little bit about your methods for conducting the study that you recently published. Like, what sorts of research did you look at?
Yes, the systematic review of the literature that we did was we did it following the what is called the PRISMA protocol and the SALSA framework that are established protocols for to write a review paper, a systematic review paper. I just want to highlight the great work of a PhD student that is first name in the paper, Amélie Poulin-Brière, and also her colleague Idriss Rezaei. They did a great work on collecting the literature and analyzing it, and then do what we call the summary, the synthesis of this data. And so our aim was to collect, to analyze all the different papers, original papers that were published with specific details on the experimental plan and specific analysis of the data. Papers that were, the aim was specifically to test an antibody with therapeutic purposes. So where we had an antibody against a protein, there are we found different antibodies used, but different targets. But the aim was really, okay, there's an antibody, we have a scientific rationale saying this protein might be detrimental for ALS. We have an antibody. We decided to test it in animal models, cellular system, or patients, and we observed then the effect of this. We had a detailed schedule of treatment, and we decided we use it. We test it in. We tested the therapeutic effect of this antibody with the analysis of then all the pathological events of that characterize ALS. And so at the beginning, we screened the main database, scientific databases, PubMed, Scopus, Web of Science, or NBase, and we found more than 5,000 publication on ALS and biomarkers. But then we had to really screen them carefully, and we use intervention keywords. So we specifically analyzed and considered only papers where there was a therapeutic intervention using antibodies, we use immunoglobulins, which are antibodies, and specifically on ALS models or ALS patients. And then we we end up with 31 publications that we grouped according to their target. so the protein that these antibodies are targeting. And we found that the first publication was in 2005, and then we had a bunch of other publications coming out from 2010 and 2020. So the majorities have been produced in the last 10, 12 years. And the most targeted protein is SOD1, which, as I said, is also the most studied one. So it's normal that we know more about this protein, so we can generate antibodies, and we can generate therapeutic approaches against that. And then we found also antibodies against other proteins that are known to be involved in ALS, like TDP, the C9orf repeats, and also other proteins that instead are involved in ALS, but where there are no mutations linked to their genes, like NOGO-A or NOGO-A, or other proteins that can be involved in the progression or in the onset of the disease, like, for example, CD40 ligand. That was just what we did in terms of methods and results that we obtained.
And in the course of this research, did you find data that would indicate whether there were any sort of targets for these kind of therapies that are particularly promising?
So from the literature review that we did, I still think that the promising targets are the proteins that are linked to ALS. So the known proteins like SOD1, TDP, C9orf. These are proteins that we know have a pathological effect in neurons and in cells. So so literature these papers prove that a treatment with an antibody is feasible. It can have a good effect. So I would push more on this protein and try to have improved the antibody-based antibodies against this protein to have a good therapeutic approach. But then, we still need to have clear biomarkers of the disease, and these biomarkers can be new therapeutic targets. So we need to work still a lot. This is my humble perspective, but we still have to work a lot on the discovery of other proteins that can be involved in the onset and progression of the disease. And that can be new targets of an antibody-based therapy.
And so, do you follow research at the ALS Therapy Development Institute? I know you have commented a little bit about our AT-1501 tegoprubart, which is the antibody treatment that we developed. But do you have any other comments or thoughts about ALS TDI?
Well, ALS TDI, who is not following the research that the Institute is doing. It's an amazing institute, an amazing initiative that is focusing all the efforts on ALS and going really from biomarker discovery, pathological mechanism understanding, and the development of treatments. It's as I said, it's an outstanding institute, and we have the proof of these amazing research on the fact that the discoveries that came out from the institute, they are now in clinical trials for patients. So yes, the antibody against the CD40 ligand is an example, and it really demonstrate how the research is well done at the institute, and how having a specific focus, which is ALS and ALS patients, helps in then making good decision in terms of science and focusing on a clear outcome, which is a therapeutic approach.
Awesome, Val. Thanks so much for joining us today.
Okay, I'm really happy to have had this interview. I'm happy. Thanks a lot for this interview.
To learn more about the ALS Therapy Development Institute's research to end ALS, visit als.net. To learn more about Dr. Pozzi's research, you can visit the Université Laval's Center for Brain Research at cervo.ulaval.ca. Thanks for listening.